an example of a real protein. And what I've got on the overhead, I'll put up in the overhead in just a sec, is a picture from my comic book. And as you can see from the comic I wrote, that was the first one I've ever done, and I have a ways to go in my artistic sensibilities, but you know, it could be worse. So what I want to talk about today are proteins. And actually, let me do this in a different color. For example, hemoglobin. And you can find this figure in today's information-packed handout. And we abbreviate hemoglobin HB, and it carries oxygen in your blood. And so, as that comic shows, there's oxyhemoglobin, which we write as HB-O2, and over here we would have deoxyhemoglobin, just plain old hemoglobin, right? And the idea is that when you breathe in, it picks up oxygen in the air, air goes into your lungs, and the oxygen from the air is exchanged by your lungs into your bloodstream, and then dropped off into your tissues. And one of the interesting little bits of trivia, it takes a minute to sort of wrap your mind around these particular numbers, but if you looked at blood, that in each cubic centimeter of blood, there's 0.2 cubic centimeters of oxygen dissolved in the water. So you can't, I mean, oxygen does dissolve in water. That's how fish breathe, right? There's O2 molecules in the water. It's not from the oxygen in the H2O. It's actually O2 dissolved in water. So some small amount of oxygen dissolves in water, and that's how fish breathe, because they get the extracted oxygen. So you could imagine using water as your circulatory fluid. And you get a little bit for every cubic centimeter. You know, 20% of this, 20% of that volume of you can dissolve 20% of that volume of oxygen in it. However, there's 20 cubic centimeters of O2 bound to the hemoglobin. All right. So what is that? How is it possible you can have 20 cubic centimeters of oxygen bound to 1 cubic centimeter, the stuff in 1 cubic centimeter? Well, the gas is dissolved in the air. 20 cubic centimeters of oxygen. Those molecules are spread far apart. In your bloodstream, it's 20 times as concentrated as the outside air. There's 20 times more oxygen attached to the hemoglobin in your blood. And there's 100 times more oxygen attached to the hemoglobin in your blood than there is in the water in your blood. So if you just use water for your circulatory fluid, it's not so great. By adding hemoglobin, you carry 100 times more oxygen per unit of blood than you do without it. So hemoglobin is a huge, big deal. The other way to look at it is if you didn't have hemoglobin, you could have about 1/100th your activity rate, which is sort of like that of a slug or something like that. I mean, even sitting in Lipke, you wouldn't get enough oxygen. So not even moving around. So hemoglobin is huge. It's a major evolutionary advantage. It allows us to lead a very active lifestyle. And so the main idea, we're going to focus in on those little squares and circles, the hemoglobin molecules in the red blood cells. Scale-wise, if you picture a red blood cell like a beanbag chair, you know, those great big sort of floppy chairs full of little beans, the beanbag chair is the red blood cell, and the beans are the hemoglobin. So it's little thousands, millions, in fact, of little beans, little hemoglobin beans packed into the red blood cell. So it gives it this flexible floppiness with all these little beans in there. Right? So it's a rough size and scale. And the comic goes on to give you some further ideas of size and scale. All right. What I want to do now is I'm going to switch back and forth between what's up on here and some computer 3D visualizations of proteins. You saw a little bit of this in lab last week, not this week. So if you look at today's information-packed handout, it has pictures from that. The images I'm going to show are extremely complicated. You couldn't possibly take notes from them. but you could take notes on the images in today's information-packed handout. So if you go past that gorgeous comic to the pictures, I'll show you what I mean. So in this software you can download if you want to play with this on your own. So that's what hemoglobin looks like in some sense. Right? What are the spheres? The spheres are individual atoms. How do you know which is which? Carbon's gray, oxygen red, nitrogen blue, sulfur is yellow. One of the notable things you don't see are any white. There are hydrogens. Does that mean there are no hydrogens there? No. You guys experienced this. I know it drove everybody crazy in lab last week, right? It didn't show any hydrogens. The reason these representations don't show hydrogens is not for the purpose of tormenting biology students. It is because that's the way this data is collected. They actually make crystals of this protein, like growing crystals of salt, you grow crystals of protein, and they shoot X-rays at them. And it turns out hydrogen atoms, being the lightest atom that there is, don't interfere with the X-rays to any significant degree. It's only the big, fat, heavy atoms like carbon, oxygen, nitrogen that actually interfere with the X-rays. And it's shooting the X-rays through them that allows them to see what these proteins look like. And basically, hydrogen is invisible. So that's why they don't show any hydrogens. There's tons of them. You just don't see them because they're invisible when this structure was determined. You could imagine where there are, which you had to do in lab. And I know that's a pain, but that's the fact of these kind of structures. So if you look at this, for the most part, most proteins are pretty boring in this view. They're basically a blob, right? And so let me show a little bit about the details of this. So this is the shape of deoxyhemoglobin, no oxygen bound. And you can switch between the two, deoxy, oxy, and the whole thing flexes, right? Why does it flex? Because the oxygen's binding inside. Why does the whole thing flex? Because it's all connected by the backbone. So you move one part, it moves all the rest. So the idea is, and actually it's probably a little easier to show this. This view shows that coily stuff is the backbone to get it out of the way. The red spidery things are the places where the oxygen binds. And so what you can see, this has got no oxygen. And if you add oxygen, those are little pink guys, the oxygens bind. So what happens is deoxyhemoglobin goes into your lungs, picks up oxygen, travels through your blood, gets to your tissues, and drops off the oxygen. So it's reversible, bind, unbind. But yeah, Levi. So if there's no oxygen, what is the red showing? There's this. So let me explain that in a second, Levi, because I can say it more clearly when I write it up. That's actually that's my next point, in